Welcome to another section of PhenoRob Graduate Talk. I will share with you some highlights of my doctoral research, which focuses on the transport and distribution of photoassimilates within sugar beet taproot, particularly under conditions of a biotic stressor and following shoot manipulations. I will first introduce us to the functional baseline of photoassimilate transport within sugar beet. So, sucrose is the major photoassimilate compound found in sugar beet. Following its synthesis in the source leaves, sucrose is loaded into the phloem and will be transported towards the non-photosynthesizing taproot sink organ. This long-distance transport is aided by the sieve elements and companion cells of the phloem. And following its arrival at the taproot, sucrose is unloaded and ultimately stored within the vacuoles of the storage parenchyma, which is located right in between adjacent cambium ring structures. The accumulation of sucrose within the cambium ring structures, the vacuoles of the cambium ring structures, determines both the physiological performance and the economic value of the sugar beet crop, and therefore a very important aspect of the sugar beet plant. However, this highly coordinated source sink system, as I've shown on the previous slide, is not static and can be affected by several factors. So, at the above ground part of the plant, herbivorous damage caused by larger grazers or tiny biting and chewing insects can reduce the source capacity of the sugar beet plant. The below ground part is also in constant interaction with soil-borne pathogens such as nematodes, which can induce feeding sites and redirect photoassimilate flow within the sugar beet taproot. Furthermore, we have to consider the activities of fungal diseases. Now, fungal pathogens can cause significant structural damage to both the below ground or above ground organs, directly interfering with the plant's ability to process and store nutrients. In addition, we have to consider vascular pathogens, particularly phloem-restricted plant pathogenic bacteria, which may gain access to the transport stream via insect vectors such as aphids or plant hoppers. So these interactions, as I'm trying to depict here, may directly or indirectly disrupt phloem subflow and affect source sink coupling. Notwithstanding, the extent to which these stresses, as I've shown here, impair carbon allocation dynamics remain insufficiently understood, and my doctoral research was dedicated to uncouple some of the effect of some of these interactions as I've depicted here. With this context established, I will outline two of the central aims of my doctoral research, which will also guide my talk today. The first one was to determine how a biotic stressor alter storage root development and carbon distribution dynamic in sugar beet. And the second one was to ascertain how shoot manipulations affect carbon distribution dynamics within sugar beet taproots. With this foundation in place, I will now move on to the first part of my talk, which focuses on the impact of a biotic stressor on the phloem submovement. One currently economically relevant vascular disease affecting sugar beet is the so-called syndrome bus research disease of sugar beet. I'll be calling it SBR just to make life easy for me. So SBR is a fast-spreading vascular disease here in mid-Europe, and the disease is known to lead to significant reduction in the taproot biomass and also sugar content, as I've shown here. And these obviously negatively affect the sugar economy. Now, SBR symptoms are quite remarkable for both the above and below ground crop organs. At the above ground, in disease samples, we see proliferation and chlorosis of leaves, as I'm trying to depict in this figure. And as observed in below ground, there is a reduction in taproot biomass, but a striking symptoms when we have a look into the slices of disease taproot samples is the brownish discolorations of the vascular bundles. Now, this disease is caused by two different plant pathogenic bacteria belonging to the so-called phloem-restricted plant pathogenic bacteria. The first one is the Proteobacterium Candidatus acinophonos phytopathogenicus, and the second one is the Phytoplasma Candidatus Phytoplasma solani. I would like to emphasize that these are so-called vector-transmitted bacteria, and for that matter, the associated plant hopper here, Pentasteroideus leporinus, is the main vector of both classes of phloem-restricted pathogenic bacteria. What this plant hopper does is that it visits healthy plants on the field, feed on them, and simultaneously transmit the SBR pathogens. Now, among several intricacies revolving around the SBR complexity is that the mechanistic understanding of the whole SBR story remains incompletely understood. And since we are dealing with a phloem-based pathogen, the trajectory of the carbon resources within the phloem and the development of the taproot, which forms the main economic value of the plant, becomes an interesting niche to study, and therefore the approach to this part of my doctoral research was to uncover carbon distribution patterns and quantify temporal dynamics of photoassimilate distribution within taproot under SBR attack. And to do this, we asked ourselves 3 questions. The first one was to determine when SBR symptoms become visible within the taproot. Moving on, we wanted to answer a question regarding how temporal disease dynamics are orchestrated within the sugar beet taproot. And last but not the least, we wanted to uncover SBR disease effect on carbon distribution dynamics within disease taproot samples. Now, to answer these questions, as I've outlined, we set up an experiment in such microcosms of, by cultivating sugar beet in these microcosms of 40 centimeter height and 9 centimeter diameter to comfortably accommodate the developing sugar beet taproot. Now, we employed one single sugar beet genotype and established 2 main treatment groups, that is the inoculated and the non-inoculated. So, to establish SBR disease in the inoculated group, we confined the plant hopper, which I've shown on the previous slide, on young sugar beet taproots for young sugar beet seedlings for an inoculation access period of 11 days, or of course under climate control conditions. And I will have to emphasize that in this study we employed only the proteobacteria, that is the Candidatus acinophonos phytopathogenicus, for the transmission assay. Now, following the transmission assay, we acquired below ground phenotypic data by non-invasive tomographic imaging. And on this slide, I will run us through how we've obtained and analyzed below ground phenotypic data from a multimodal imaging approach. As I indicated earlier, we're interested in the below ground structural development following SBR disease. And for that matter, we employed the plant dedicated vertically oriented magnetic resonance imaging platform to acquire root phenotypic data structurally. And for functional carbon allocation dynamics, we employed tracer transport technology and positron emission tomography to monitor the pathway of recently fixed photoassimilates. So what we do here is that we label the whole shoot of the sugar beet with 11C, the radioactive carbon isotope 11C, in the form of 11CO2. Now, what that basically means is that we supply the carbon 11 as 11CO2 to the shoot of sugar beet plant, following the assumption that this labeled photoassimilate will be incorporated into the recently phased photoassimilate. And for a period of 150 minutes, we are able to track or monitor the movement or the pathway of the 11C within the sugar beet taproot by positron emission tomography. Now, the next step is to reconstruct the data obtained from both the MRI and PET modalities into 3-dimensional images. And from these 3D images, we are able to either single-handedly extract structural information or combine both MRI and PET for structural and functional carbon distribution dynamics. And the data obtained from this step will then be analyzed quantitatively. So in this experiment, we were able to image all the plant samples, but for the MRI, but for the PET, we had to dedicate 6 plants for the PET acquisition due to the current tropotivity that we have. Now, let's have a look into some results. On this slide, I'm showing you temporal and spatial analysis of a healthy and a diseased taproot. So we've analyzed the 2D slices at different spatial positions. That is the upper, mid, and lower sections of the taproot for the control and also for the disease samples. And we also did this temporarily, that's during the disease progression period. And here we observe some qualitative changes in cross-sectional tissue structural organization, which was exerted by the SBR pathogen. In fact, in the disease samples, we observe some differences in cross-sectional tissue arrangement. You might appreciate it more when we have a closer look, and therefore let's have a look into the upper slices at the last imaging date for both the control and the disease samples. If you agree with me, we observe clearly delineated anatomical structures from the central core to the storage parenchyma and also for the cambium rings in the control samples. But we have seen a change in MRI signal for vascular rings and for storage parenchyma, and a pattern of broadening and interruption of vascular structures as shown here. All these indicating alteration in structural integrity of the storage route upon SBR disease. Moving on, we quantify taproot volume as a direct measure of storage route development. And to quantify the taproot, we first generated a binary image from the MRI based on an image intensity threshold. And subsequently, we removed the fine root structures and soil samples by just applying a median filter. Now, finally, we manually selected the base of the taproot, and the remaining taproots, as shown in orange here, defined the volume. And from this, we could observe the temporal development of the taproot. So on this figure, I'm showing taproot volume. On the vertical axis and the horizontal axis, I'm showing this after inoculation. Of course, the control is rendered in blue and the disease samples are rendered in red. Here, we have observed that the presence of the SBR pathogens caused a progressive reduction of taproot volume from 49 days after inoculation, and this continued until 63 days after inoculation. As I mentioned briefly in one of my earlier slides, sucrose accumulation within the cambium rings determine the physiological performance and economic value of the plant, and therefore we analyze SBR effect on ring width development. And for ring width analysis, we contoured manually an estimated ring width of the 4 inner rings, as I'm trying to show here, by making sure we excluded the central core. So the distance between the central core and the 4 inner rings define the ring width, and we have observed some differences in ring width development. As I'm showing on this figure, ring width on the vertical axis and this after inoculation on the horizontal axis. Again, control is rendered in blue and disease samples are rendered in red. We could observe that the presence of SBR caused significant reduction in ring width development already at 42 days after inoculation, and this progressed until the last imaging day, which was 63 days after inoculation, all indicating a reduced internal taproot ring width development by the presence of SBR pathogen. Now, let's have a look into the next part of the resource session, which explores tracer transport distribution within the taproot, which has been achieved by a combination of both imaging modalities. Here, the MRI is rendered in gray and the PET is rendered in colored. So the colored part of the image will represent the PET and the gray part represent the MRI. And on this slide, I'm showing you a video depicting the tracer distribution within the taproot. So we have observed that in the control samples, the tracer filled almost every part of the cross-sectional area, indicating an even distribution of Levin's photoassimilate resource. But for the disease samples, we observe a pattern of a sectoriality in resource distribution, where part of the taproot receive little to no photoassimilates. And I will have to clarify that this part that received little to no photoassimilate were in congruence with the sectors that showed altered structural organization per the MRI modality. So from this, we could have an indication of a form of a sectoriality in resource distribution following SBR disease incidence in sugar beet taproot. So we move further from this spatial identification of sectoriality to have insight into on the temporal scale, where we had an indication of sectoriality already at 42 DAI, but was more pronounced at 49 DAI and progressed until the last imaging day, which was 63 days after inoculation. And therefore, we could confirm that sectoriality indeed was induced earlier than 63 days after inoculation. Moving on, we determine temporal heterogeneity in tracer transport distribution within several slices of each taproot. Here, I'm showing tracer heterogeneity as per the vertical axis and days after inoculation on the horizontal axis. Again, control is rendered in blue and the disease samples are rendered in red. And here, the circles represent heterogeneity values for each taproot measured, and the blue and red lines represent their linear regressions for control and disease samples respectively. Now, we observed that the heterogeneity increased during the course of the experiment by a factor of around 3 for each day in the disease samples, and for the control samples there was no such pattern observed, and all these indicated a progressive uneven carbon distribution, which is consistent with an impaired phloem subdistribution upon the presence of SBR pathogens. Let's have a flashback here. Earlier on, I highlighted that the biotic stress aspect of my doctoral research was dedicated to answering 3 major questions. The first one was to determine when SBR symptoms become visible within the taproot. Indeed, you will agree with me that the SBR-induced alterations on Cambrian ring structures was already prominent at 42 days after inoculation based on our MRI modality. Also, we wanted to know how the temporal dynamics of SBR disease development, SBR affect taproot development. And here, we could also have insight that the SBR induces a progressive reduction in taproot ring growth and volume development. Last but not the least, we wanted to know how SBR disease affect carbon distribution dynamics within the sugar beet taproot, and it was clear that SBR induces a temporal increase in heterogeneity in carbon resource distribution. Now, to further investigate sourcing coupling in sugar beet, and the next session of my talk will examine how shoot manipulation influences carbon distribution. Of course, this was also in line to deepen our understanding on the observations made from the previous chapter, which I introduced you to briefly. Now, the overarching aim of this chapter was to ascertain how shoot architecture and manipulation of shoot tissue will affect carbon distribution dynamics within sugar beet. And this session was also dedicated to answering 3 very useful questions. We wanted to know how a specific leaf distributes its carbon resource within the taproot. Again, we wanted to know if shoot manipulations by either excising one leaf or shading will affect the carbon distribution dynamics within the storage root organ. Last but not the least, we wanted to know if different form of manipulation will trigger different form of carbon distribution pattern within the sugar beet taproot. Once again, same technologies, but a bit different labeling approaches, which I will introduce you briefly to. So first of all, we labeled the whole shoot of a sugar beet with our 11C and monitored the distribution pattern. Following that, we singled out one source leaf, labeled it, and followed its distribution pattern within the taproot sink organ. And later on, either excised or shaded the leaf that we previously labeled and perform a whole shoot labeling and monitored the distribution pattern within the sugar beet. I will want you to grab this labeling approaches, as it will influence how the results is being rendered. So, as I indicated, we did a whole shoot labeling, single leaf labeling, and again a whole shoot labeling before inducing our manipulation. And here we could observe that indeed from the whole shoot labeling, we observe of an intact plant, we've observed homogeneity in resource distribution in both either morning or by repeating the measurements in the afternoon. But when we labeled one single leaf, there was a striking effect here. And what is striking here is that we observe that a specific leaf supplies its recently phased carbon resource to just a specific sector of the taproot. And indeed, the sector that received the 11C was in congruence with the architectural position of the labeled leaf. And therefore, we were convinced that there is a sectorial transport of carbon from an individual leaf to a certain taproot zone. So, what happens if we excise this source leaf, which previously supplied its photoassimilate to this sector of the taproot? In fact, the assumption was that this sector will be void of recently phased photoassimilate, but indeed that wasn't the case. Following the shoot manipulation by excising the source leaf that previously supplied its photoassimilate to this taproot section, we observed that when we remove this source leaves, the distribution pattern was unaffected. Indeed, there was a uniformity or a homogeneity in resource distribution after following the shoot manipulations, following the distribution patterns from 0 to 6 hours, 24 to 30 hours, and 48 to 50 hours, thereby confirming that uniformity in carbon distribution is maintained post-defoliation. But what happens when we don't excise this leaf but render it transport inactive by shading it? And therefore, we perform the shading experiment. Also, similar labeling approaches: whole shoot labeling of an intact plant, single leaf labeling, and repetition of whole shoot of an intact plant before manipulation. And here we observe similar pattern of sectorial distribution, which we confirmed in our previous experiment. And what happens when we shade the leaf that previously supplied its photoassimilate to this taproot sector? And here, 0 to 6 hours during shading, we observe that a sector of the taproot was deficit of the 11C, where the 11C signal was reduced for the sector where the shaded leaf previously supplied its photoassimilate, and therefore we could confirm that shading induces a form of a localized 11C deficit within the taproot. But continuous monitoring 24 to 30 hours during shading, we observed that this deficit began to be, these sectors that were deficit of 11C began to be filled to with 11C to a good amount of 11C intensity, which resembled the distribution pattern of an unshaded plant, and therefore we could conclude or assume that there is a compensatory redistribution of 11C to restore carbon distribution upon shading. Of course, this observation was of interest for further investigation, and therefore we investigated the distributing pattern further. And on the next upcoming slide, I will show you the temporal distribution patterns or the temporal intensity patterns of 11C for prior to shading and 0 hours into shading and 24 hours into shading. On this slide, I've shown you the maximum intensity projection, and in the upcoming slide, I will show you the temporal intensity projection just for these 3 selected samples. So, during the course of the experiment, we observed that 55 minutes into the experiment, for whole shoot labeling of an intact plant, the 11C was arriving earliest within the inner first to 4 cambial ring zones and began to spread towards the outer rings to the periphery, which was prominent at 30 and 90 and 135 minutes. And therefore, we could conclude that indeed there is an early arrival of 11C within the inner taproot zone, which is followed by spread towards the outer and the peripheral zones of the taproot. But how does the distribution patterns looks like for a shaded plant? And here we observe from that early minutes, around 55 minutes of during the measurement, there was a striking sectoriality where a sector of the taproot was deficit of the 11C, and this pattern began to be reduced, or the sector began to receive 11C during the course of the experiment. And a similar pattern was also observed 24 hours of shading, where there was an initial striking sectoriality followed by increase in 11C signal intensity around the section which was void of 11C. From this, we could confirm that indeed there is an initial heterogeneity in resource distribution followed by a compensatory redistribution to achieve resource distribution homogeneity. In that sense, the taproot acts as a unifier and either redistribute its 11C to sectors which are void of 11C. On the last flashback for the day, earlier on I highlighted that this part of my doctoral research was dedicated to answering 3 questions. The first one was to determine how a specific leaf distributes its recently phased photoassimilate resource. And here, you agree with me that a specific leaf supplies its resource to just a sector of the taproot. Also, we wanted to know how should manipulations affect carbon distribution dynamics. And here, we have established that homogeneity in resource distribution is prioritized post-manipulation. Last but not the least, we wanted to know if different manipulations, that is either pruning or shading, will induce different carbon distribution pattern. And here, we observe that while shading induces a prior heterogeneity and a compensatory redistribution of 11C, the uniformity in 11C is maintained post-defoliation. I will summarize the major findings of my doctoral research into 3 thoughts. And first of all, we are certain that the non-invasive tomographic imaging approach enables early detection of SBR-induced anatomical alterations, where we observe that we could detect SBR-induced alteration earliest at 42 days after inoculation, especially from the MRI modality. Also, from the manipulation experiment, that is the shading and pruning, we have clearly established that the taproot acts as a unifier to redistribute its photoassimilate resources, and this is aided by vascular interconnections, which is allowing the flow of photoassimilate to sectors with little or low 11C signal. But the presence of a vascular destructor like SBR pathogens can override these compensatory mechanisms, thereby affecting the sink capacity of the developing taproot. Together, the findings of my thesis bring forth mechanistic understanding of carbon distribution dynamics in sugar beet taproot and how biotic stresses may impair 11C distribution within sugar beet taproot. But of course, there is more room for improvement, and for further mechanistic insight, it will be beneficial to determine the regulation of sucrose transporter genes, especially under the cases tested in this thesis. Also, the detailed knowledge of the ultrastructure of the sieve element is critical to any discussion of the mechanisms of translocation in the phloem, and therefore, future studies should employ high-resolution microscopy of the phloem architecture. From a phenotyping perspective, it will be beneficial to link above and below ground mechanistic traits, as this will provide a holistic overview of functional and structural relationship existing between above and below ground plant organs. In a broader picture, the structural and functional mechanistic traits established in this thesis can be and should be used as a basis to judge susceptibility and select superior genotype that can benefit future breeding programs. Also, our MRI and PET tomographic imaging approach should be extended to other vascular dysfunction pathosystems. Thank you for your attention. I'll look forward to your feedback, comments, and questions.